Hundreds of thousands of researchers around the world are working to improve life and address imminent threats to humanity. Often their research ends up in the scientific valley of death in the form of publications and patents that never see the light of day. That is about to change. Welcome to the Lab to Startup podcast, hosted by Naresh Sunkara, founder and executive director of the Berkeley Postdoc Entrepreneurship Program at the University of California, Berkeley. This show has two main goals: share the stories of those who have successfully founded startups based on their own research, and highlight resources needed to help those aspiring to launch startups in the deep tech space. Whether it's electric cars, vaccines, addressing climate change concerns, or possibly establishing life on other planets, Naresh and his esteemed guests want to help scientists, engineers, faculty, and researchers bring their innovations to market. Learn more and subscribe today at labtostartup.com. And now, here's Naresh.
My guest today is Dr. David Kirn, the founder, CEO, and chairman of Ignite Immunotherapeutics, which was recently acquired by Pfizer. David is a physician-scientist and a serial biotech entrepreneur with over 25 years of experience in the industry, with a focus on viral vector-based therapeutics, including gene therapy and oncolytic virus cancer immunotherapy. I previously had David as a guest on my show talking about 4D molecular therapeutics, where he was the founding CEO and took the company public. Because of the long and successful experiences in the biotech industry, we will start the conversation by talking about acquisition models in the pharma and biotech industries, and then talk about the origins of the idea behind Ignite, how the relationship with Pfizer was initiated in a novel build-to-buy model, details of how the relationship worked, benefits of working with big pharma, and how startups should think about such relationships. I'm really looking forward to learn about this new model and hope you enjoy this conversation. David, welcome to the show. I would like to start by congratulating you on making it to the Medicine Maker Power List for 2022, which is a list of the people making the most impact in the drug making space in the category of advanced medicine, which is a very truly deserving recognition for you. Also want to congratulate you on your startup, Ignite Therapeutics, being acquired by Pfizer, the topic for today's conversation.
Excellent. Well, thanks for having me.
I thought it'll be an interesting place to begin by talking about history about pharma biotech industry, because you've been there for about 25 years now, on the topic of acquisitions and the models that existed. That way, we'll set up the stage for what happened previously, where Ignite went, and what the future might look like.
If we think about business models for biotech companies, the traditional business model is to raise cash through equity financings to build a company to a point where you have value and you can start to do pharmaceutical partnerships, eventually a public offering or an IPO. Most companies, if they get that far, eventually get acquired by a large pharmaceutical or a large biotech partner. The number of companies that make it all the way to become fully integrated, entirely independent, multi-billion-dollar companies is pretty rare. Most companies get acquired. That's been typically the model through which companies get acquired, go through multiple rounds of serial venture financing, do some partnerships, and then at some point, it's usually a company that you're already partnered with on a program decides that, hey, at this point, it's more cost effective for us to own the whole thing than to continue to partner and pay out these milestones and royalties. That's sort of the historical way this normally works.
Thank you for setting that stage. That is really helpful for people to get a good perspective. Let's jump into the topic for Ignite. Tell us about what you were trying to solve at Ignite Therapeutics and the solution at a higher level.
On a technical level, the problem we were trying to solve is that cancer immunotherapy, which has great promise, does not effectively work in the majority of human cancer patients. We've seen great strides with cancer immunotherapy. It can be potentially curative, but the majority of cancer patients either are not amenable to cancer immunotherapy, or if they respond, it's transient and survival is still limited by their cancer. We believed we had a technology with oncolytic viruses to use those therapeutics in such a way that they not only attack and debulk the tumors in the body, but also help to stimulate a patient-specific immune response to their cancer in a way that could be combined with the traditional cancer immunotherapy of immune checkpoint inhibitors. It was really coming up with a broadly applicable, patient-specific, off-the-shelf stimulant for combinations with their cancer immunotherapy in their pipeline.
That is fascinating. Let us dig a bit deeper into that subject. Maybe you can give some more background of the amount of work that went into oncolytic viruses at your end to where you thought you had to start a company, and probably touch upon the subjects like what are the existing technologies and how your solution was different or better.
The concept of using a live virus to attack and destroy cancer cells is more than 100 years old now. From some of the earliest days in the early 1900s, when viruses were first discovered and cultured in the lab, people noticed that they were pretty good at blowing up cancer cells. As the virus would replicate and be released from the cell, it would lyse the cancer cell. So very early on, people had this idea that viruses could be used for cancer. And over the subsequent 100 years or so, there were multiple attempts periodically that are reported in literature of people trying to use different wild-type viruses, because this is pre-genetic engineering days. These are all wild-type and present in nature, trying it on different tumor types in the lab or trying it in some animal models, and even a number of clinical trials in patients where wild-type adenovirus or measles virus would be injected into tumors directly in patients. Bottom line is they generally saw pretty good safety, but lack of significant durable efficacy in those patients. There's this interesting idea that's been around for years and years. What happened in the 1990s is by that point, we had enough of an understanding about the genetics of each virus and the genetics of the human tumors and the genetic engineering techniques that we could manipulate the genome viruses. When that all came together, then we could actually do this in a much more intelligently designed and rational way, where we could now say, okay, based on my knowledge of the viral genetics and how the virus takes over a normal cell and the way that a tumor cell evolves from a normal cell, I can take those things together and I can start to delete viral functions that are necessary in a normal cell but are expendable in a cancer cell, because the cancer cell is already changed in the way that the virus would want it to be to replicate and multiply. The first group that took something like that seriously forward and into the clinic was my team at Onyx Pharmaceuticals, where we took an adenovirus that was altered in a way that it was dependent for its replication on the inactivation of p53 in the tumor. The virus would be turned off in normal tissue, but it would be turned on and replicate and express its genes in p53 deficient cancers. P53 deficiency is probably the most common genetic abnormality in cancer, so this had very wide potential applicability. We went into the clinic and went from concept to phase 3. We treated over 400 patients on more than 15 different clinical trials. That was really the first attempt to do this in a serious way. We got it to phase 3. And then at that point, I went back into the sabbatical looking for more of a systemic virus, because the adenovirus we were using had to be injected directly in the tumor, which limited its applicability to a fraction of all human cancers. I worked on technology with vaccinia virus in academics at Imperial Cancer Research Fund in London and at Oxford University during a sabbatical to come up with an intravenous systemically active oncolytic virus for cancer. And that was the genesis of what went into my company, Generex. We took that concept all the way to phase 3, and we sold that company to essentially a private equity group in Asia. Taking that concept to another level is when I started Ignite. And that's what led to the build to buy with Pfizer.
It's been over 100 years and it's still not to the point where we can use them as medicines. Part of the problem might be the viruses, I believe. Hard to predict the behavior?
If you look at the totality of the data, and again, there are probably more than 10 different virus species that have gone into human cancer trials. So everything from adenovirus to herpes virus to vaccinia, measles, VSV, polio, reovirus. It's really remarkable the number of viruses have gone in. If you had to summarize the totality of the experiences, the safety was remarkably good across almost all those trials. And the efficacy was relatively limited as a single agent without adding exogenous transgene payloads to the virus to also get additional potency. Viruses on their own in vivo in tumors are not generally potent enough to give you what you want, to give a durable response, a durable complete response, and major survival advantage. And that's probably due to the fact that viruses did not evolve to really destroy tissue and move quickly. Most of them may be beneficial to be more stealth for the immune response, but grow more slowly. They don't necessarily want to blow up cells when they spread. And then, of course, we have the antiviral immune response that kicks in a human that's going to dampen that effect. If you think about the three-dimensional structure of a solid tumor, there are a lot of barriers to a virus spreading: fibrotic tissue, normal cells, etc. We realized, look, well, in a dish, an oncolytic virus, a single virus can wipe out the whole dish. That's not going to happen in humans. And so now, how can we make it work better? There's two fundamental ways. One is make the virus itself better by adding in new transgene payloads, for example, that can cause direct lysis of the cells or cause an anti-cancer immune response or put a bullseye on the cancer cells so the immune response can recognize it. You can think about prodrug activating enzymes, so then you can give chemo and activate the chemo locally. So there are a number of ways that we could use we use the term arm the virus with additional transgenes that make it work much better. That's one general approach. And the other general approach is combining with standard therapies in intelligent ways. So we found, for example, that the oncolytic virus on its own in head and neck cancer might give 5% responses. Chemo might give 20%, but the combination was giving us 70%. So it was apparently synergistic. We discovered that viruses induce certain cytokines like tumor necrosis factor, which are potent chemosensitizers. Likewise, with immune checkpoint inhibitors, you could imagine synergy with the immune response induced by the virus and the transgenes in the virus, plus the checkpoint inhibitor might be synergistic. So that's generally where the field has now gravitated towards: better transgene payloads and better combinations.
You mentioned something about the virus, oncolytic virus, wiping out the cancers in a plate. That is something of a lot of interest for me because industry is complaining about we don't have good in vitro models or good animal models. What, in your opinion, might be a better approach for these in vitro testing assays before we go after the clinicals?
There's not going to be one model that's perfect. I think what we try to do is use a number of different models that have different strengths and weaknesses and try to use that to get the best predictive value from what you're going to see in humans. For example, a traditional nude mouse human tumor xenograft with a cell line that's been grown in the lab for 50 years. That system, it has no antiviral immunity, no anti-cancer immunity. It's got a cell type that probably has nothing to do with what you'd actually find in the human today. So probably not very predictive. You can move to human tumor explants that are more recently taken out of humans in cultures, so they haven't been passaged for decades in the lab. Those are so-called PDX models. They may be a little bit more predictive. You can go and say, no, I need an intact immune response, so I'm going to use a mouse tumor model. That has the advantage of an intact immune system, but the downside is a lot of these viruses have differential replication and spread in a mouse tumor versus a human tumor. So you've got that complexity. Unfortunately, I don't think there's any one model that's perfect, so you have to cobble together several and see whether they all tell you the same thing.
Absolutely. There's so many problems there. If I may ask you, other than your program, is there a program with oncolytic virus that you're impressed by in any of the company or in the field? And what is impressive about that?
If we think about oncolytic viruses, we want something that replicates quickly and spreads efficiently in tumors. And you want something that has a big transgene carrying capacity. So you can add in these transgene payloads that arm the virus. We love vaccinia because it has all that, and we believe it can go intravenously very efficiently. We showed that in a clinical trial that we published in Nature. Another virus that has great potential is herpes virus, for example. It's got very rapid replication, very large transgene encoding capacity. There is an approved product. The only oncolytic virus that's approved is a herpes virus expressing GM-CSF that's used in melanoma. The downside with that program, and they've got a follow-on program in a company now called Replimune. The original company, BioVex, was purchased by Amgen, and this Replimune is now a publicly traded company developing a next-gen. Again, the downside with that is they're not able to give it IV, so they have to rely entirely on the systemic immune response, which is pretty tough. I think that's certainly got promise. There's another company called Oncoris out of University of Pittsburgh that has a promising herpes virus that they're trying to make that go IV. And if they could accomplish that, that would have some promise. I do think that the idea of using a directed evolution approach to create novel viruses that have features that you want, that has great promise. We're doing that with AAV, not for cancer, but with non-replicating AAV at 4D. And we did do that at Ignite with vaccinia, and that showed great promise. Applying directed evolution to get even better viruses is another important component here. There's a company called Cioxis that has taken an adenovirus and evolved that. That's another example of a promising company and technology.
I worked a lot with the herpes virus during my postdoc, so I have some good insights into that space. Let us shift gears and try to talk about the business, how we built it. One of the first things that comes to your mind when you want to launch a company is funding. When you're thinking about Ignite, what are the thoughts you had about how to fund it? What are the options you had? And how did Pfizer come into picture?
I'd like to kind of break things down to their core to help people understand them. When you think about financing a company, you can either get grants, which is essentially money for free to start a company in a lab, and that's fine. But once you're at a lab, it's hard to generate enough cash to do something meaningful with that. But there are small business innovation and research, SBIR grants and STTR grants to get you started. This is an industry where if you're successful, you're going to have to raise hundreds of millions of dollars. I think we've raised now 500 million since inception, and we're just now in phase 1-2 trials with 5 products. Still got a long way to go. How can you raise real money that will be differentiating and allow you to push things forward? It's really a pharmaceutical partnership where you get cash and research funding and milestones and eventually royalties in return for them getting the future product rights to that product. That's one way. And the other way is to sell shares in your company, but not the rights to any particular product. You're just selling shares in your entire company. Anyone who owns a percentage of your company owns a percentage of everything that's in there, all the products. Those are the two fundamental ways. And then there are blends of all those. You can do a pharmaceutical partnership that also includes some equity financing in there. But generally speaking, those are the two big categories. They both have strengths and weaknesses. The advantage of going with a pharma partnership is you're not diluting your ownership in the company because you're not giving them shares, which means you're not losing control of the board of directors, which makes some critical decisions because you're not having to put one of your investors or two of your investors on the board. You're also not losing control of key decisions like when can I sell my company? When can I go public? Those are all things that are defined in favor of the shareholders who come in and invest in your company. The longer you can avoid that, the longer you can retain that control. The downside of a pharma partnership is obviously you've given away a lot of the value. The earlier you do it, the more value you give away, but you've given a lot of that potential value in that product away. And if you do that too much, let's say you only had 1 or 2 products, they were partnered for royalties. An investor is going to look at you and say, why would I invest in you guys? You don't really own anything of value. You gave it all away to this pharma partner. You got money for it. I want you to have products. It's a double-edged sword. You have to be very careful that if you do pharma partnerships, that you leave more than enough for yourself, because that's going to then allow you to raise money from investors when you're ready to do so. And the ultimate exit is typically, even after an IPO, is eventually an acquisition, as we said in the beginning. It's an acquisition by big pharma or big biotech. If you've given away too many product rights, they're not going to be interested in you. They're not looking for a royalty stream. They need to put their salespeople to work and leverage what they built there to generate a revenue stream from products. Those are the two traditional ways, and that's why there's a tension between the two of which is better for you. When Pfizer came knocking on my door after I'd sold my last company and said, hey, we love what you're doing. Would you like to do something new with us? And they proposed a partnership. I said, well, that could work if you just get a couple of transgene payloads and I get to keep everything else. And they're like, no, well, we really want something broad. Can't we do all vaccinia with you? And I said, well, not really, because if you do that, then you essentially own my company and I won't be able to raise additional funds because you guys own the rights to everything. I'll just be this zombie company that can't raise money, can't develop its own products, and is just waiting for a royalty from Pfizer. I said, I don't think you want to buy us today because it's so early. And they're like, no, we want to make sure it works. That's where I came up with this new idea of saying, what if we bridge the gap? What if we say, You're going to essentially partner with us. You're going to give us cash. We're going to agree on the development program. We're going to do some things. You guys can work on your end and do other parts of the development program. And after 3 years, if you like what you see, you buy us, and this is how much you're going to pay. We had to do an analysis of what a company at our stage 3 years in the future, if it was successful, would be worth, and align on that agreement with Pfizer upfront. That way, we get to sell the company at an early stage before there's any additional dilution by VCs. So that's a win for us. They get to pay less because there's not all these VC middlemen that they have to pay off. It's really a win-win. And the advantage for them is we were building exactly what they wanted, and they got insight into the research the whole way. So it wasn't like they had to rely on our word for it. They were working on these reagents in their own hands, these products in their own hands. For us, it was great because we knew we were building exactly what our customer wanted. We didn't have to raise money and build products for 3 years and then go out and see if people wanted to buy it. We knew this is exactly what they wanted as long as it worked. It allowed us to be very, very focused. We didn't have to talk to other partners. We didn't have to raise money. I didn't have to worry about building a board of directors or doing press releases. We could just focus on getting the job done. We could focus on what we were really good at, which was the innovation, the product design, and Pfizer could focus on what they were really good at, which was manufacturing, high throughput animal model testing, and cancer immunology.
All these decisions look very complicated to me and complex, and assuming it was not easy. Can you give us a sense of the kind of people that were helping you come to that decision?
I was fortunate that when I jumped into the industry from my academic faculty position at UCSF, I jumped into a very early stage company. I was probably employee number 10 or something. I got to really learn from my 6 years at Onyx. It was a great group of VCs and management behind that company. I had a front row seat. That was very helpful. And then I'd done this before with my company, Generax, as well. I'd had a lot of experience to see the pros and cons of these other things. The other people who've been very, very helpful is Alan Mendelsohn, who's now, unfortunately, the late Alan Mendelsohn, but was really the godfather of biotech corporate law, was an excellent advisor over the many, many years. He and Judith Haskell at Latham on BD and financing and everything else. And then Mark Robinson's a very close friend of mine from my undergrad days at UC Berkeley. He's now arguably the number one M&A banker in all of life sciences. Just by chance, actually, he was a history major at Berkeley. He's at a company called Centerview. He's at B of A in Merrill Lynch before that. Just a top banker. And so he is deeply, deeply, deeply experienced in how to build a company so you eventually can get acquired if that's the direction you want to go. So I think it was people like that over the years have been really helpful to me to really understand how to structure these things and what the pros and cons of different funding approaches were.
Speaking of Pfizer, can you share some high-level terms and conditions from both the parties, from you and from Pfizer, around the work to be done, the board, the timelines that help you get into this relation?
When you think about negotiating a business agreement, it's really important, I think, to start with what are people trying to accomplish. Don't even talk about quote unquote terms. The second you put terms on a term sheet, it becomes a competition. What I like to do is say, look, what do you guys want? Why are you doing this? What does success look like for you? And really understand that deeply before you start talking about terms, because there's a lot of different ways to structure a deal that are win-win, that work great for both parties, but you have to understand what each party wants and needs. It started with me asking them a lot of questions. Why are you doing this? Well, we believe in cancer immunotherapy, but we think checkpoint inhibitors aren't going to be enough, and we want something that's off the shelf. I'm like, great, we can deliver that for you. But they said, we don't want to pick one transgene payload or two today because we might discover great ones in 2, 3, 4, 5 years. I'm like, okay, I get it. If I did a partnership with you today where you had to pick the cytokine today to put in there as the definition of the field, you're uncomfortable with that because you don't know that you won't discover something in the next year that's much better. So I say, I get that. You need to have the ability to have a series of products here as you learn more and more. Once you understand that, you say, okay, I can deliver that for you, but let me tell you my problem. What are my pain points? If I give you a deal where you say you get everything for the next 20 years, how am I going to raise money? I just gave you everything. I might get some money to run my company for a year or two from you, but how am I going to raise my next round? And they said, oh, okay, I get that. You can either buy me now, but I don't think either of us wants that. You're going to give me such a low price. It's not worth it for me. And you're not going to want to make a big bet until you see the data. So how can we create a situation that benefits us both, where you can have it all, but I can actually get a meaningful exit here? What you do then is you ask the question, okay, well, what's a long enough time to decide whether they want to buy us? Okay, 3 years. We got a research plan. 3 years should be sufficient. As long as we deliver X, Y, and Z, then they have to make a decision. If we don't deliver and we're delayed, then we have one more year. They can stretch it out by a year, so we have more time to get more data. So that was addressing one point. We then had to agree on a price. If we're successful 3 years from now, what is a company at that stage worth? And so you go to comparable public companies, any private companies that you can get the data on. You can do a bottoms up net present value of the products, and you triangulate, negotiate around a price there. The next thing was agreeing on the research budget. How much money do they have to put in to fund the company? And then the next step was, okay, what if we don't stay together? What does that look like? For them, they said, well, we want to be able to get out and not have to buy you, but we want something in return for all this funding we gave to you. So can we walk away with some shares? And I said, well, I'm willing to give you shares, but I can't give you shares that have any special rights for voting, because then you could control my company. And if I want to get sold to your competitor, you can block it. So I said, okay, you can have shares, so you have a financial reward, but I can't give you any control. They said, fine. And then what I said to them is, look, I don't want to get to the end of this, and now I'm out of money. And you come back to me and you say, well, I don't want to buy you for X. I want to pay half of X. Then I'm over a barrel. I'm very, very vulnerable. So I said, if you don't buy us for the full price, you have to give me an additional 1 year's worth of cash to run my company and give me time to go raise money from an investor, because I can't be at your mercy by having run out of money, because then I know your business guys are going to come back and say, yeah, we're not going to buy you for X. We're going to give you half of X or whatever. Where am I? I have no money left. Pfizer's walked away. How am I going to raise money from the next guy? That was really important for us to say, you have to give us 6 months notice whether you're going to buy us or not. If you're not buying us, you got to put in another year's worth of cash. So then I knew I had a runway to go raise more money. Now, fortunately, they bought us, but if they hadn't, we were well set up. And the last thing I'll say before I take your next questions is, you never can align perfectly on the price. One way to bridge it is to say, okay, fine, give me this much up front. If the product works out and hits another important milestone, then you give me another tranche to me and my fellow shareholders. That's what we aligned on. Is there was an upfront payment, and then there was a milestone payment when the product that we designed and developed, when that went into the clinic, Pfizer had submitted the IND and had an open IND. They would then have to pay us another tranche, such that if for whatever reason our new product was too toxic in toxicology models or had some other problem and they couldn't take it forward, they wouldn't have to make that payment. But if they took it forward, they would. And so fortunately, the product got into the clinic. So we got that additional payment that helped to bridge the gap between our two positions on valuation.
So here we are, you have your agreement in place. Can you walk us through what your experience was working with a big company once you started working with Pfizer? How did they complement, if any, and what was that experience like? Were there any notable obstacles that you had to overcome in that process?
First of all, at a high level, it was amazingly good, which I did not expect at all. When you have a traditional partnership where the partner's one step removed, where they're not doing the tests themselves, inherent mistrust can develop. Can I believe this data? Oh, there's a delay. They're not being honest why there's a delay. What's going on? It's just human nature. If I have an internal program that I'm running myself versus a partnered program, it's at least twice as much work and at least twice as long to get anything done. Because you think about it, you have to decide on everything internally. Then you have to package it, present it to your partner. Then you have to negotiate with them to agree on what to do next. It's incredibly slow and painful, and it can be a huge distraction for your company. Not always, but that can happen. In this case, it was very interesting. It was all totally transparent. We were working together as one team. So what we let them do is large scale stuff that they were really good at. Manufacturing, analytical testing at large scale, There were certain changes we were trying to make in the virus to make it behave in a different way that required some very specific, expensive equipment that they already had. So we had them do the high throughput stuff. You can imagine if you want to test 10 different transgenes in a viral backbone, and each one has to go into a couple of mouse models to understand what's happening. You can imagine how large the animal studies were. And so we let them do that. You guys are experts on animal models and cancer immunotherapies. You guys can do all that. What we focused on was the viral delivery vehicle design, engineering, and characterization, because that's what we could uniquely do well. At any given time, I think we had 10 people on it, but they probably had 20 to 25 on it.
Were there any obstacles? Did everything go as planned? Number one, I'd be surprised. But if there were obstacles, how did you overcome? And what is that collaboration like? Because they had the skin in the game. They wanted you to succeed. What did that look like?
One of the great things about this program was if we succeeded, we were going to succeed together, which meant for them internally in their company, it was very attractive to them personally. Now, in contrast, if you do a partnership and a small company does all the work, sometimes the pharma partner, the people there get resentful. Why are they getting all this money? I have a better project. Why are you funding this? If we bring this in-house, it means more resources for them, less for my project. By flipping that on its head and saying, no, this is your project too. If this succeeds, it's not an Ignite success. It's an Ignite and Pfizer joint success. That's going to look much better for them internally, and they're going to have ownership in it. And that's just something I didn't anticipate. I didn't plan that, but that's how it worked out. And I was really very, very surprised at how integrated it was, because I've been through so many partnerships where it was tense and difficult. This one was seamless. Everything we said, they were testing in their own hands at the same time. And so we felt more confident in the results when somebody independently confirmed it and vice versa.
This is such a collaborative model. What has taken industry so long to come up with this? I just want to go down that thread of built-up cell model. Give a sense of what those conversations were like in terms of who was at the table talking to you and how long did those conversations last before you came to a conclusion?
It was me on our side because it was sort of my concept of the company and I was the founder, support from my attorney. On the other side, it was originally their head of business development for oncology. Eventually, since this was such a new structure, I got kicked up all the way to the top of the head of all business development at Pfizer. The two of us negotiating this and creating a new model, which was a lot of fun. A guy named Bob Smith, who's just a great business development leader at Pfizer. So it was the two of us. And because it was such a new model, and it took me a while to get them to understand why a traditional partnership would not work, I'd say all in it took a year.
I think that's pretty fast.
It did not feel fast at the time, but I guess it is. To make that work, you needed a couple things. First of all, you needed some very strong internal champions at Pfizer who could convince bureaucracy to do something radically new. So that was Karen Hughes and James Merson, and eventually Bob Hollingsworth and Claire Lisu, the strong internal champions who could make this go pound the table with their business and legal teams and say, look, we got to do this. Number two is you have to have a long track record as a leader in a field to get one of these things. I think the fact that at that time I've been doing it for 25 years, I don't know that anybody had developed as many different oncolytic viruses and taken them into clinic and published on them and built companies around them and gotten them to phase 3. So I had a track record. They could say, okay, you're a brand new build to buy. You have no data, but you have a track record. It's not something you could do straight into your career. Those are two big things. Internal champion, something that's of high importance for the company so people are willing to do something different. And then you have to have a very long track record of leadership in that field to be able to pull this kind of deal together. The other way to do it that has been done more in maybe devices and things, but a few times in pharma, is to do your traditional financing rounds and build a data set that's compelling enough over your first 3 or 4 years and then convert to build to buy. That's doable too. It's just that you're going to have more investors, more dilution because you had to raise those rounds of financing. And then the pharma company now has to negotiate with the VC at the table, which is going to be a little bit more difficult in terms of how much money they want. That's another way to do this. If you don't already have a track record, is go out, raise some money, and build something that's valuable enough that a pharma says, yeah, I'll do a build to buy for the next 3 years after that. And that's another way.
Having those internal champions is such an important, big deal for any of these relations. I've been hearing the story again and again, and your experience, that goes a long way. As you mentioned, for somebody, an early stage founder who might not have any of these, either the relations or the experience, can we establish some mechanisms? This is hypothetical. If you want the rest of the industry to build on this model, do you have any recommendations for an entrepreneur?
I would say that you're going to need the champion and you're going to need that track record. And so if you don't already have those two things, then you're really going to need to take some initial financing, equity financing, and grants and build enough of a valuable data set to be able to then do something like a build to buy instead of a partnership. It's doable. You just need to invest 3-4 years of work likely. If you had a world-class academic who everybody knew was a leader in the field and they had generated a patented technology that was ready for prime time and going into a company, that would work. Company 47, I think it was Irv Weissman from Stanford, had this CD47 signal on cancers. And I think that was one where it was pretty close to a build to buy, or maybe they did one round of finance and they got acquired.
We definitely need these new models. I'm so glad you succeeded. This opens up hopefully doors for other entrepreneurs in the future. I want to slightly shift gears and talk about something you mentioned during our last conversation about monetizing science in terms of translating it to a business. Can you elaborate on the point for scientists and founders in the space, or academics in the space for that matter?
When I teach, I love to boil things down to their core, because then you really understand it. There's all this noise around business models and this and that, and it all sounds very complex. People get very confused as to what it is. They start to think, oh, I raised money, I earned a profit. Like, no, you didn't. You gave up your company in return for cash, and now you have to build something worth much more of that cash, or you're actually losing. What I try to do is boil it down. If you ask the question, what is a business model in biotech? In my mind, it's how do you monetize science? When you're monetizing science, you're primarily monetizing data. Sometimes you can monetize capabilities as well, but generally, even to convince people that your capabilities are valuable, you're going to need a lot of data. You're essentially monetizing data. Entrepreneurship and business, it's very simple. There's got to be a customer, and that customer has to be willing to pay more for what you make than what it costs you to make. If you really boil it down, that's what it is. And I know in biotech, it's like all this fancy technology and all these fancy investors and blah, blah, blah. But at its core, you have to remember, that's what it is. When you monetize that science as your business model, you have to have a terminal payout that gives people a return on their investment that was worth the risk and time it took to get that. So that's an acquisition. Or an IPO where eventually your trading volume is high enough that they can eventually trade their shares. It's liquid enough that they can trade their shares without pushing the stock price down. That's the ultimate. If you stay independent, then what is that revenue stream that matters? And that's either you're selling your own products for profit, which is, again, very rare to get to that point as a startup, or you're getting some sort of royalty or a kind of royalty called a profit share, where essentially it's a kind of royalty. So that's the bottom line. That's why, for example, with this build to buy Pfizer, I'm like, look, if you come in and you do this broad partnership, then I have no way to get that terminal exit because no one's going to buy me and no one's going to invest in me, which means I can't go public. So I can't have that terminal exit. If I sit around and wait for 15 years for you to commercialize this product, if you do, the royalty is so small that it doesn't make up for the time value of money of waiting for that. If you boil these things down to very simple principles, you're monetizing data. And the way you're monetizing it is eventually an acquisition, IPO, or some sort of royalty or profit share or ownership of a product, which takes a long, long time. When you raise money, people again think, oh my God, my company's successful, I raised money. Well, no, it depends. What did you give up to get that money? Was it a good deal? Are these good investors that you want to be with? Do you want them on your board? You could do a huge raise and it could absolutely kill your company forever. It might be terminal. You might take a relatively small raise, but it sets you up perfectly for long-term success. You have to look at these things and realize that fundraising is not in and of itself profit and is not success. What it's giving you is resources that you can use to build data that has more value to someone else than the money you took in. So if you raise $300 million, you better use that to create data sets that are worth more than $300 million. Otherwise, you're losing money on that investment. It's just very interesting if you force people to actually think these things through and say, oh, okay, that's how this thing works. It helps crystallize people's thinking.
Maybe that's a nice way to segue into another thing we talked about how pharma is the customer, big biotech is the customer. How can we best frame this for startups or founders or people who are trying to form these companies? The fundamental fact that for most startups in the drug discovery space, big pharma biotech is your main customer. And how should that customer discovery be programmed? How do we plan that?
Absolutely. That's always been a point of mine is the ultimate customers, big pharma or big biotech. Why is that? Well, if you have to raise tens of millions and eventually hundreds of millions of dollars, and somebody else has to buy you for more than what you put in, how many organizations are there out there who can pay that kind of money? Basically, it's the top 15 or 20 biopharmaceutical companies would have that kind of money that they could deploy to buy you. Now, you can say, well, that's not totally true, Dave, because I could go public and sell my shares. Yes, that's true. However, when you talk to professional investors, the ultimate win for them is get you public. So now you have leverage with the pharma, and then pharma has to pay more for you than if you were private. Eventually, their big exits, their 10x, 20x exits, almost always publicly traded companies that are acquired. If you don't have the ability to get purchased by a pharmaceutical company because you've done some partnership on your lead asset so no one will buy you, then it's very difficult to get investors to invest enough in you to get you public and be able to sell those shares. Whether or not you sell the company ever or in that time frame, the possibility that you could is, I believe, necessary to raise the kind of money you're going to want to raise to be highly successful.
I agree with you 100% because I deal with a lot of early stage startup founders at UC Berkeley, and I keep telling them, don't try to go from A to Z. You'll probably die getting there. Plan better, A to E, E being an exit. How do we get there and build that data? That's going to be of most value. Before I get to my traditional closing questions, one last question I have is, how can founders be better prepared to work with big pharma in terms of what are the general mistakes, common mistakes that you have seen when they make partnerships, whether it is timing? I don't know what you've seen, if you have any observations.
When I think about a business development partnership, the headline that these biotechs want is, oh my God, it's $1 billion deal. It's like, no, it's not. You got $50 million up front. Maybe 2/3 of that or all of that is earmarked to do the work to develop the pharmaceutical partner's product. So you could see that if I gave pharma a whole huge therapeutic area that was my most promising, I got cash in the door, but I have to put that cash towards their product, not my own, where I owned it 100%. That could be a value crushing deal. Now, I might announce it as, oh, I got $1 billion deal. Everyone's like, oh my God, $1 billion. Well, no, you just crushed your company. What you have to think about, the most important thing is, number one, keep that field as narrow as possible. So you still have plenty of things that you can do that you own 100%, and therefore you can raise money off of. And also you want other fields to be able to do other partnerships with other companies in the future, especially at a higher valuation later on after you've had more proof. You also want to make sure you structure in a way that you still could be acquired by somebody else. You don't want to do such a broad deal where they have step-in rights on your second and third products and they own the first one. Well, no one's ever going to buy you. These investors are very sharp. They're going to look at that deal and they're going to like, no other pharmaceutical will buy you. So now there's no bidding war, which means you don't get bid up to where I can make a 10-fold or 20-fold return on my money. The best you can do is 1 or 2-fold, and that's not worth it given the risk. Keeping that field narrow is critical. I think upfront cash that you can use for your own products is critical, or even selling equity at a premium is better than getting a bunch of cash, but then having them say, yeah, but you have to use that cash on our product. They're not even giving them up. They're using you as a CRO and you're not making a profit. keeping the field narrow, keeping the upfront cash high for your own uses that you can use to build your own company and your own products. And then the third thing is it's the exit revenue that really matters to people who know what they're talking about. What is that royalty? What is that profit share? If you can retain an opt-in right in the US, we say, okay, my option, I can co-develop, co-promote and retain more of the profit in the US, even if I have to pay up to keep that. I think if you can keep the US rights or an option to US rights, it helps a lot because that's what investors are really going to want to see. All the intermediate things of, I got $10 million for filing a BLA and $20 million for a BLA approval in the US, another 20 in Europe. Those add up to big bio bucks, but those don't change anything you do with the company. They don't change your valuation. They don't change the way you run the company. They're pretty worthless. So I would focus on field long-term, trying to retain as much value at the late stage as possible, including trying to keep some US rights or opt in for US rights. That's very valuable. Maximize the cash up front and don't sweat so much the development milestones.
Such important observations. Thank you for sharing those, David. Now on to my traditional closing questions. The first one, are there any lessons you learned from mistakes you might have made in this process with Ignite that others should or could avoid?
I think I would have preferred to have more control after they bought us. I wish I still had more control over the publications and things like that because those publications help you with your next company. And so that's been more of a struggle. So I think I would have asked for a little bit more control, a little more access to the clinical data after they acquired us. So I had more insight to what was going on. I certainly am a consultant for them, but it's definitely arm's length. So that's probably one thing.
Cool. The second question is, are there people that played a significant role in your Ignite journey that you want to mention?
Karma is real. If you treat people well, it comes back and benefits you in a virtuous cycle. I would say Karen Hughes is somebody where when I was starting my company, Generax, back in the early 2000s, I collaborated with her when I had a consulting business. I had a consulting business essentially to bring in capital to feed my family. Because I needed a day job, I needed money, and then I could also deploy the extra to starting Generax. She was somebody I collaborated with at a company called Cell Genesis, which business-wise was kind of a failure, but scientifically was amazing. We had a great working relationship. I really helped her out on a number of things as a consultant. And then it was years later that she approached me after a talk that I gave at a Keystone conference. She was now at Pfizer, and she was the one who said, hey, let's work together. I think she was absolutely critical, because if she hadn't walked up to me after that conference, that deal never happens. James Merson and Bob Hollingsworth were the guys at Pfizer who sequentially ran cancer immunology and were great product champions. Claire Lease was the kind of scientific head internally who took over after Karen Hughes moved on somewhere else. And then ultimately it was Michael Dolston who made the decision to acquire. Those are definitely people I love to highlight, and co-founders of mine like Teresa Jenke and Jenny Holt and Sharon Tetlow and people like that were also essential.
David, this has been a fascinating conversation. I learned a lot. I'm sure my audience is going to learn and appreciate all the lessons that you have shared. Thank you so much for your time and all the lessons.
Thanks, Ramesh. It's great to talk to you today.
Thank you for listening to this episode of the Lab to Startup podcast. You can find links to the resources and programs mentioned in these episodes, connect with Naresh, or subscribe to this show at labtostartup.com.